Hello students. So here let us learn about the dihybrid cross. So when we talk about dihybrid cross, it is nothing but the study of the inheritance of two different character or two different traits we can say. So when we understand the inheritance pattern of two different characters at a single time, we use the term, right, dihybrid cross. So to understand this particular cross, let us take an example. We will take two characters from where we'll use the same plant that Mendel had used, that is the garden pea, also known as the Pisum sativum scientifically. Right. So we're going to take two characters here. So what are the two characters? One is going to be the, let's say, the seed shape. We can take the seed shape as one character. So in the seed shape, we are going to have the dominant form, right? What will be the dominant form? It is going to be round, and the recessive form as the contrasting character, that is wrinkled. Okay. Now the second trait, let us talk about the seed color. So here the dominant trait of the seed color is going to be yellow, not the green. Many times students confuse here, and the recessive trait here is going to be the green. So accordingly we have to take the alleles, right? So for the dominant one we are going to have the capital R seed shape, and for the recessive one smaller. Similarly, for the seed color we are going to have the capital Y for the yellow color and small y for the recessive green color. So you all know students that whenever we talk about the dihybrid cross, for example, you have already learned about in the monohybrid cross that we always start with a homozygous parent, that is pure plants. So here let us start with parents and they are homozygous for both the traits. So when we talk about homozygous for both the trait, that means the plant here must have round and yellow traits. So when we talk about round and yellow traits, the genotype here is going to be capital R, capital R, capital Y, capital Y. Now you might think that why am I taking two letters, two alleles for each? Because we know that we are talking about a diploid condition. That is why we have to take both of these in pairs. We know that alleles are going to occur in pair. So one of the plant here is going to be capital R, capital R, capital R, capital Y, capital Y. So this is going to be our round and yellow seed. Okay, this is one of our parent. Now the second parent that we will talk about or that we will take into consideration is going to be the recessive one. So in this case we are going to have the wrinkled green seed. Okay, wrinkled green seed, and for that we are taking the small r, small y, small y. Now obviously these were the condition in case of a somatic cell, but we know during reproduction they are going to form the gametes, and we have already learned that the gametes are going to be haploid in nature. Because they are haploid in nature, out of these two, right, for each of the character we had how many? Two alleles. Now out of these two alleles we are going to get only one of the allele here. So from this R and R, because they are the same thing, we are going to get capital R, and here we are going to have capital Y, again we will get only one, that is capital Y. So this is only one type of gamete that we are going to obtain from this parent plant with round and yellow seed. On the other hand, for the wrinkled and green seed, the plant containing that, we are going to again get only one type of gamete, that is going to be smaller, small y. During the next stage, that is they are going to fertilize. So when fertilization is going to occur, right, they will give rise to progeny, and this progeny here is going to be a combination of the gametes here. So it is going to be capital R, then there is the same trait, small allele, that is a recessive allele, small r, and capital R, Y, small y. And this one, my dear student, is going to be the F1 progeny. So what do you mean by F1 progeny? So when we say F1, F1 refers to the first filial generation. Okay, remember this. So this F1 refers to first filial generation. So here the expression, phenotypic expression, what will be? As per the law of dominance we have learned, it is going to be round and yellow seed. Okay, again we are going to get the round and yellow seed. So this one actually also known as the hybrid. Okay, we are getting the hybrid seeds. So as you can see here, there is something missing, right? Here you cannot see any plant that is going to have this wrinkled and green seed. Here all the plants that were produced only produced the round and yellow seed. So there was this RRYY, or this wrinkled and green condition. Actually this was missing. Okay, it is actually missing. So the question is, are they actually lost? Right? Are they lost? Right? Are they actually not going to be inherited? So to answer this, again we have to go for the F2 generation. Now in the F2 generation, right, again we're going to start with the parent. So F2 parent, what is F2 parent? F2 parents are nothing but the F1 progeny, right? So capital R, capital Y, small y, and they're actually selfed, right? It is called selfing with the same one, that is capital R, capital Y, small y. Now here again they are going to form the gametes. Okay, here they are going to form the gametes. Now the thing is, it is not as simple as we saw in the previous case. Here what happened? Very simple. We said only one type of gametes are going to be produced, capital R, capital Y, or here in this case it was small r, small y. But what happens in this case is slightly different. What is that? Here we will get four type of gametes. What are those? Capital R, capital Y, 1. Okay. Then capital R can go with small y, second. Third, the small r can go with capital Y, and the fourth one, small r, small y. Right. So these four we are getting from here, the first parents. Similarly, for these also we will get the same outcome, that is capital R, capital Y, right? Then capital R, small y, and then we are going to have small r, capital Y, and small r, small y. Now the question is, how are we actually getting these? So to understand this, okay, I'll just make you very simple. I'll try to make it very simple. So we are going to have capital R, capital Y, small y. So at a point, at a single time, we know that the capital R is one of the allele of the trait. So capital R can go with capital Y. So we can say this, capital R, capital Y. So the capital R can go with also the small y. So capital R, small y. Similarly, the small r can go with capital Y, and the small r can go with small y, because here the alleles are going to be independent. They are not going to always go with one particular allele of the other trait, but instead they can go with any of the allele of the alternate trait. That is, what we are saying is, the dominant allele can go with the dominant allele of the other character that we are talking about, or it can go with the recessive allele of the other trait that we are talking about. Okay, so this is how we are going to actually get these gametes, right? So once we get the gametes, okay, let us stop here. Once we get the gametes, then we have to go for the F2 progeny, right? So how we will get the F2 progeny? So to get the F2 progeny, again we are going to go with the Punnett square. Okay, remember we are going to go with the Punnett square. So to make the Punnett square, let's start the process. Okay, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. On this side also to write capital R, capital Y, and capital R, small r, capital Y, and small r, small y. Okay, so why did I write in this? these four, let's say from one of the parent, we are going to have the gametes here. Let's say it is from the female side, and this is from the male side. Okay, and okay, let me change the color. Okay, so here we are going to have the gamete first. In the first line, we are having all the gametes. So first we have to make the square, Punnett square, and then we'll go on about filling all of these. So now what you have to do is you have to start filling it up. How do you fill it up? Right. So as we were saying earlier, here capital R, capital, these are one set of gametes and this is one set of gametes. So when they're going to fuse during reproduction, during fertilization, they will give rise to the progeny. So here capital R, capital R, capital Y, capital Y. So again next here capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Similarly here capital R, capital R, capital Y, small y, capital R and capital R, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. So here we are going to have the capital R, small r, capital Y, capital R, small r, capital Y, small r, small r, capital Y, and here we are going to have small r, capital Y, small y. Then you are going to have capital R, small r, capital Y, capital R, small y, small y, small r, capital Y, small y, and small r, small y, small y. So this is what you're going to have in the Punnett square as of now. So this is what we have written as a genotype, right? We have written all the genotype. So once we write the genotype, then we have to write the phenotypic expression. So what are the phenotypes here? So here we are going to get round yellow. Okay. So let us find out what all are going to have the round yellow condition. Okay. So I'm just going to copy paste, right, all the places we are going to have the round yellow. So you can see here round yellow. So this is also going to be a round yellow. This also is going to be a round yellow. Now this is also round yellow, and now I think this is also round yellow. Then we are going to have round yellow. Here also you're going to have the round yellow, and here you're going to have round yellow. Now how many we have? 1 2 3 4 5 6 7 8 9. Okay, we don't have any more round and yellow. Second phenotype that we're going to get is let's see here, round and green. Okay, so again let's find out where all we have the round and green condition. Okay, so here we're going to have the round and green condition. Here we are going to have the round and green condition. Okay, so the next one that we have here is going to be the wrinkled, right? Round is over, wrinkled yellow. Okay, so again how many wrinkled yellow? Let's find out. Here we have one. Now wrinkled yellow, here you have wrinkled yellow, and here also we are going to have the wrinkled yellow. And finally what you're going to have is going to be the wrinkled green, right? It is a wrinkled green. So these are all the genotype and phenotype that we see in the dihybrid cross. Now let us find out ratio. Just like monohybrid cross, what are the ratios here? So first here no need to worry about genotypic cross in your standard. So we'll just talk about the phenotypic ratio. So when we talk about phenotypic ratio, as we had discussed, first thing that you have to do is you have to go on about writing the phenotypes. What are the phenotypes? So here we are going to have 4 different phenotypes as written in the 4 different colors. I'm going to use the same 4 different colors. So one is going to have the round and yellow seed. Okay, that is one of our phenotype. The second one is going to have the round and green phenotype. Okay, then we are going to have another phenotype that is called the wrinkled yellow phenotype, and finally we are going to have the wrinkled green phenotype. Okay, that is completely homozygous recessive. Now we have to count how many of these are falling into each of these different phenotype to find out the ratio. So round yellow, So we are going to have, I'm going to strike this so it will be very clear. 1, 2, 3, 4, 5, 6, I'm sorry, 5, 6, 7, 8, 9. A total of 9 are round and yellow. Now how many are round and green? 1, 2, 3. 3 are round and green. That is heterozygous for one of the trait. And how many are wrinkled yellow? So 1, 2 and 3. 3 are wrinkled yellow. And how many are wrinkled green? That is always going to be only 1 for homozygous recessive condition. So here the phenotypic ratio, as you can see, for the dihybrid cross is going to be 9:3:3:1 for the round yellow seed, round green seed, wrinkled yellow seed and wrinkled green seed respectively. And any of the trait that we are talking about is following, if they are going to follow the Mendelian inheritance pattern, they will have this phenotypic ratio of 9:3:3:1. So this is how, my dear students, we are going to have the inheritance of the two genes of two traits at a single time, and the phenotypic ratio turns out to be 9:3:3:1 in all the cases. So this is how we solve the dihybrid cross or the dihybrid ratio. I hope you are clear with these concepts. Thank you all for watching this video.